Okay, I think I'm the only thing standing between you and your dinner. Yeah, I'd like to, we stumbled on a discovery that there were little membrane-bound packages being excreted into the space between the mating junction that Jennifer had just been describing. So I want to tell you about what we've learned about these so far. First, I had to learn the different kinds of extracellular microvesicles that there are in the world. This is a really complicated literature. On the one hand, there's these things called ectosomes. And these are, you can think of them as like viral shedding particles. That's when the plasma membrane pinches off and it's taking with it some cytoplasmic contents and exiting into the outside world in the extracellular environment. Alternatively, and I had to learn about these, I saw these in Lydia Bright's talks and some others, multivesicular bodies. These are late endosomes, and they tend to in-pocket, and often they're taking ubiquitinated cargo and packaging it into little membrane intraluminal vesicles that get swallowed up in a larger vacuole of sorts. These are multivesicular bodies, and I had seen these in the EM and sort of didn't know what to make of them at the time. But these things, if they fuse and go through exocytosis, you can see how they also would release membrane-bound compartments into the extracellular environment. And people have been, these are all the rage apparently. There are a lot of people studying extracellular microvesicles. They're packaged with proteins, they're packaged with RNA, and people are beginning to discern signaling functions for them. So just a few examples. People find them in some parasitic roundworms. They have a distinctive shape. They kind of look like flattened discs. And in some of the parasitic roundworm cases, these extracellular microvesicles are affecting their host. They're actually suppressing the immune response so that they're better able to invade. In humans, cancer cells have been shown to secrete these, and these EMVs have been demonstrated to kind of condition their surroundings to better enable the cancer to take hold. This is a shout out to Clive Wilson. I don't know if he showed up or not. But here they found that in Drosophila, the testis produces EMVs that coat the sperm, and when they're delivered, they actually affect the female's behavioral attitude. She's less likely to mate again. So there's a lot of really interesting signaling going on. In something closer to our heart, Chlamydomonas, it's my understanding that the male produces little EMVs, sheds these little exosomes or ectosomes, and that they actually can carry a lot of the protein that's involved in preparing the cells for mating. So they've been implicated in cell signaling. They're found pretty much everywhere people started looking. So it's kind of exciting that we came across some evidence for them possibly in mating Tetrahymena. I'll point out the mating junction. Again, Jennifer did a nice job of introducing it. This was one of Judy Arias's early sections, and it's a glancing section through the mating junction. I just love it because it picks up the mating junction and the pores that Jennifer talked about. So here, these are individual pores, cytoplasm from one side, cytoplasm from the other. And it's a rare microtome blade that catches that kind of nice surface. More typically, the EM catches a cross-section. Here's the pore, here's a pore, reminding you this is all extracellular space between the two cells. And there's a little pocket with some little vesicles inside. I don't know. It didn't impress me much the first time I saw it either. I started looking back through some of the older pictures and literature. Judy had taken this picture, and again, it's neat. There's pores, and then there's this thing. It looks like a tooth. And inside, but this is extracellular space, and again, some little vesicles. It looks like junk. I couldn't help thinking of two kids kissing with chewing gum. It's like, ah. Here's a really older picture from Jason Wolf. Again, capturing this junk in between. Why would you study that? Nobody would. But they're there. And we found more and more pictures of them. Our gallery began to grow. And so my first question was, well, are these pinching off, or are they multivesicular bodies fusing and disgorging a bunch of intraluminal vesicles? Courtney Azello, you've heard her name several times. She was an undergrad, I think, at Boulder. and has gotten more and more responsibility for running the EM facility. She's brilliant at it. She did serial sections for us, which in some ways give you information you can't get from the beautiful tomography. There's a time and place for everything. But there's the mating junction of a wild-type Tetrahymena, and this is the same section going deeper and deeper, and you can see, I hope you can see, there's a neck to this little vesicle pinching off into the outside space. This next series is again, serial sections. There's going to be 3 pores, one here, one pore here, and we'll get another pore here as we go deeper. And you'll see that these little vesicles are going to show up in between. Pink arrows are going to mark the pore, new pores, and again, you can see, I'm going to go back and forth through it a few times. There's pores, and then there's this little package of vesicles inside there. Again, maybe not too impressive. Occasionally, we would catch a section that showed the neck of these little vesicles, and it's really compressed. So you can imagine this thing is kind of being pinched off in an extraordinary way. Well, when you've seen about 20 of these pictures, you start to think they might be something. They might be something interesting. So how could we study their contents? Again, this harkens back to this lovely collaboration with the Clark Lab and with Donna Cassidy Hanley and Jennifer, where we had such fun looking at this hap2 mutant that makes no pores and the wild type that does make pores. And the thing is, the hap2 mutants, you can knock them apart by just breathing on them practically. So simply centrifuging them, they'll fall apart. A gentle centrifugation causes the pairs to fall apart. And I thought, well, these would be great if they only had some of those microvesicles. Well, it turns out they have giant vesicles trapped in the little extracellular space between here. So we thought, well, this is at least where we can start. You can knock them apart very gently without disruption. We might be able to do differential centrifugation and harvest these things. And in fact, we did. This is my undergrad, Sasha Dimitrenko. A lot of people think he looks a little bit like Tom Hanks. So we harvested these rather enormous vesicles, and they look like little flattened frisbee type of things from the hap2 and from the wild type. And Sasha even got some from vegetative cells. So they're not just confined to mating cells. They also are produced at other times in the cell cycle. I got to tell you, the work was brutal. It takes days to grow up enough cells. You're dealing with liters of mating cells that you reduce to maybe a volume of 50 microliters of these little vesicles. And it didn't take long for Sasha to go buggy. So he decided he needed a vacation this summer and went off east somewhere for his health. Anyway, following a hunch, I wanted to look and see whether my old friend, the Bcd pattern mutant, might have some of these things, and it was one of those rare hunches that pays off. I'll tell you more about the Bcd pattern mutant, published the same year as the Drosophila Bcd mutant, so we got stuck with the same acronym. But ours stands for broad cortical domains. But again, here's a pair of Bcd mating cells, and they produce just piles of little vesicles in the extracellular space. So I thought that was, and this is my new favorite picture of the Tetrahymena mating junction. It's a thick section that we're going to do tomography on. I've promised it to Sasha for his hard work. He's going to do the modeling with the tomography on this in January. We'll be bringing 10 more kids out to Boulder to do this. So, but here you can see the kind of folded membrane junction with the pores, and here in this one little pocket, a whole pile of these little vesicles again. So what's inside these things? Well, Sasha harvested these things, and we did proteomics on them. We found 377 different proteins inside them. Some of the categories were kind of interesting. There's membrane trafficking proteins. There are proteins associated with the proteasome and ubiquitination pathways. So we're starting to get the idea, maybe they're like intraluminal vesicles in that they are capturing ubiquitinated cargo to be exported. Maybe this is the garbage system. Maybe we're throwing them out of the cell in little membrane-bound packages. One other group of proteins were Rna Binding Proteins. And one of those in particular was kind of interesting. It had a fair assemblage of the Argonaut homologs, the TWE1 and TWE2 proteins. And I'm taking caution. it's quite likely, it's quite possible that our vesicles are simply collecting cytoplasm randomly. They're sampling the cytoplasm at the time. These were harvested at about 2 hours into mating. This is getting into meiosis. It's when the mic genome is broadcasting all those little RNAs, the scan RNAs. We could simply be harvesting these RNAs. We could simply be picking up scavenging some Argonaute proteins, some TWE1 proteins from the cytoplasm. So I don't want to get too excited about them. That said, Sasha went ahead and measured the RNA in our little pellet of vesicles and found an abundance of RNA. So we shipped it down to St. Louis, to Washington U, and they did some RNA-seq. They found a lot of RNA, a lot of different sizes of RNA. So we looked, again, playing the hunch, we looked at the small RNA. I think it was less than 100 base pairs. And we got this rather remarkable result. This is just in. I don't even know how to think about this stuff yet. But we had 24,000 hits. I remind you the genome is about 27,000. It does look like if you were broadcasting your entire genome in little bits of RNA and our little vesicles were picking them up either randomly or in a directed fashion, they'd be sampling pretty much the whole genome. So it's that's in the back of our minds as we're thinking about this. Maybe these vesicles are removing membrane. Maybe they're also, everyone keeps talking about how the scan RNAs go against the macronuclear RNAs, and the ones that find matches are eliminated, but no one talks about how they're eliminated. Maybe they get packaged. At any event, we've got, the thing that we're solid on is we've got structures. We've got these little compressed discs and vesicles that are spit out into the extracellular space. They're rich with proteins. They're rich in RNA. We just don't know what they do, and we're looking for a function. In kind of following the lead of these other places, we're really keen to see if we could demonstrate a signaling function. So we thought, well, maybe they're involved in pairing. You know, Chlamydomonas seems to secrete vesicles that stimulate pair formation. At this point, Mark Lee stepped in and took over Sasha's place of working 3 days without sleep. And he harvested them, and we'd throw them onto cells that were co-stimulating. There was no effect. Sasha did an experiment where he centrifuged away all of the vesicles and tried that medium, and it didn't slow them down at all. So adding vesicles, removing vesicles, nothing seemed to affect pairing dynamics. Pairing went on oblivious. We also looked at when cells had already formed mates and were deciding, are we going to commit to post-zygotic development, or are we going to abort? Some of you know this decision point that pairs make. So we threw the vesicles on like mad. Mark Lee worked like crazy, giving me more of these vesicles. He'd work for days, and I would take them and squirt them on, and they wouldn't do anything. So we don't see a signaling function yet. I think we have maybe one more experiment to see. But we are left with two possible roles for these microvesicles. One is to help excavate membrane materials from the mating junction. I'll remind you, we get these little membrane protrusions. This is supposed to be one cell membrane, the other cell membrane, the extracellular space. And the protrusion punches through to form a pore. And the little protrusions could continue and could serve to help excavate, because these pores that start about 100 nanometers do broaden. They do widen, and so these pores do get larger. And where does the membrane go? This is a possibility. Maybe this process is helping you to excavate the membrane as these pores get larger. Another possibility is that they could be involved in eliminating the scan RNAs. We see the Argonaute protein in there. We see a lot of RNA. We have a huge representation of a transcriptome inside these vesicles. So the last thing I wanted to go through quickly was just to remind you of our two mutant phenotypes. HAP2, which makes giant ones, and BCD, which makes an abundance of these little vesicles. Very quickly, I'm just going to touch on these. It looks like HAP2 fusogen might be necessary for these EMVs to pinch off. And I'm not sure whether that's direct or indirect, but I'm picturing this thing as a balloon that keeps blowing up and doesn't get to stop. So membrane keeps being fed into these vesicles, and instead of pinching off, they seem to be just ballooning up in the HAP2. Now, what about the BCD mutant? I wanted to tell you a bit more about that one. Quick, let you know, BCD, broad cortical domains. I did my PhD work 30 years ago in Joe Frankel's laboratory with this mutant. It's fun to come back to it. A normal cell has one mouth, BCD has multiple mouths. A normal cell, 2 contractile vacuole pores, whole string of contractile vacuole pores, extra cytoprocs. Every cortical organelle we looked at was multiplied, was broadened in the mutant. And I had been thinking, what could be causing that? Well, 30 years later, I had no more clue. But this EMV phenotype has given me a new way to think about it. So just very briefly, 2 models. You could imagine that when you're going to specify where a new mouth forms, you might have to send little marks out to there, marking the spot where you're going to build a platform to build an organelle. And it builds the mouth. Whereas in the BCD, maybe we have a hypertrophy. We add more markers and therefore end up making more mouths. One way to think of it. The other way to think of it is that there's already lots of markers and we have to clear them. We have to excavate material that will then allow you to assemble an organelle. And perhaps we have an overactive excavation process allowing you to build multiple organelles on the platform there. And I got to say, watching the BCD cells kind of hyperactively producing vesicles kind of has caused me to kind of favor the possibility that the BCD, the EMV phenotype, is perhaps involved in an overactive excavation of materials to clear the regions where cortical organelles are going to form. I will say that when we've looked at EpiC, it's a cortical a protein that forms in the epiplasm layer. It clears out from where the mouth is going to form. So it's kind of interesting. There's the old BCD mutant, multiple mouths, multiple contractile vacuole pores. Here's the wild type, single mouth, single pair of contractile vacuole pores. Kevin Cook always accused me of drawing those in with a felt tip pen. These are old silver stains. I want to thank the Boulder group. Mark Winey has kind of been a host to St. Olaf for years. Courtney for her wonderful work, and Thomas Giddings with his. The Cornell group, again, Donna and Jennifer and Ted were very generous in bringing me in on that collaboration. U of M, Mass Spec, WashU, RNA-seq. And these guys, there's Sasha, the real Sasha. Adrian don't have a picture for. Joe Frankel. There's Mark Lee, who doubles as a, I don't know, something. And just a shout out to Claire and Aaron and Sabina. I hope you all got to see their posters. They've been a terrific crew this summer. I'll stop there.